And today I'm going to talk about the CD8 T cell differentiation after COVID-19 vaccination. As you know very well, T cells are very important for the protection against severe COVID-19 disease, especially against variant virus. And during the pandemic, so once I suggest that the T cell-oriented strategies are needed for the effective controlling of the COVID-19 pandemic, especially in the era of variant after Omicron. And with this background, the T cell importance, I'm going to talk about today about the T cell phenotype differentiation. In my talk, I'm going to talk about two different small topics. And first one will be the durability of memory T cell response after vaccination. In this first part, I'm going to talk about the stem cell-like memory T cell differentiation. In second part, I'm going to talk about the effect of repeated mRNA boosting vaccination. Then in the second part, I'm going to talk about the terminally differentiated T cell. But let me start with the first part. So during the pandemic, we once studied what happened in CD8 T cell differentiation after 2-dose vaccination. As you can see here, so we obtained, the blood sample, the longitudinally, and then we stained the pentamer staining. Actually, the staining was done with the ProImmune pentamer. And so actually, from the pre-vaccination, we can see very nicely the priming of the CD8 T cell and then the boosting after second vaccination. And then these are summary slides from the subject. As you can see here, this is log scale. After first vaccination, there's some increase of the T cell frequency. And after second vaccination, again, the more boosting. But we found that during the 6-month follow-up period, the frequency of tetramer positive cell significantly decreased during the 6 months follow-up period. And then we want to know, so what is the CD8 T cell durability, how it is regulated. So in the tetramer gating, we look at the effector memory, TEMNA, or central memory to such kind of differentiation status. And then we found that in the effector memory, like CCR7 negative, CD45RA negative, it is after acute stage, it is increased and gradually decreased during the follow-up period. And TEMNA is we regard them a terminally differentiated one. It is gradually increased. And TCMs also. And then so we found that the stem cell-like memory T cell, it is gradually increased after second vaccination. And then what's stem cell-like memory T cell? As you know very well, the human memory T cell can be subdivided to the central memory, effector memory, or terminally differentiated TEMNA in that way. And then double positive cells are regarded as naive cells. But if you try the SARS-CoV-2 tetramer, pentamer, then even after vaccination or infection, you can find certain cells are located here, naive cells. But they are not naive cells. So they look like naive cells, but they co-express CD95. Real naive cell, they don't express CD95. So it has been known that naive cell-like, but they express CD95, they are stem cell-like memory T cell. Compared to any other T cell, they have the maximized self-renewal capacity. And also they have the multipotence to be differentiated to any other cell type. In that way, this one is required for the long-term protection, protective T cell immunity. And then previously, in the SARS-CoV-2 convalescent patient, once we showed that after salting the stem cell-like memory T cell, we stimulate them with the peptide epitope. And then we showed that they are very highly the proliferation capacity. And also, after the starting point was stem cell-like memory T cell, but after the stimulation, they became not only stem cell-like memory T cell, also central memory, effector memory, and TEMNA. So in this way, we showed that multipotency. And also they showed in this in vitro IL-15 stimulation, they have a high self-renewal capacity. And then, let me go back to our vaccination story. And then we defined that the durability. The CD8 T cell durability in this way, in the very simple equation, we divided the MH class of multiple frequency at 6 months by the MH class of multiple frequency at the peak time point. And then what determined such kind of durability? Very simple answer. We found that. So this one is the durability. is a nicely correlate with the stem cell-like memory T cell generation after second boosting vaccination. And then, what determined this the stem cell-like memory cell generation? Age. So not so big cohort, but nicely we could find that in these the young age people, they have the much more the stem cell-like memory T cell generation, but old people, they have the less generation of stem cell-like memory T cell. So in my first part, I showed that this the T cell differentiation status depending on the subject age. They are showing the different type of differentiation, and this different type of differentiation can lead to the different durability. So in this way, we once published that the stem cell-like memory T cells are very important for the CD8 T cell durability after vaccination. But then, the first part was a brief part, and my second part, I'm going to show you what's the effect of repeated mRNA vaccination. So even in this time, well, for example, my mother got every year the SARS-CoV-2 vaccination, boosting vaccination, mRNA vaccination. Then what's the effect? We don't know yet. Then especially in the early pandemic, we got every 6 months mRNA vaccination, so those even first dose. So what's the outcome of this, the repeated mRNA vaccination on the CD8 T cell differentiation? To know this effect, we recruited some cohort. So this cohort, they got the first vaccination, second vaccination, third vaccination. Then we compared this after second vaccination and after third vaccination. So this one is a representative fact plot for the Pentamers staining, and then this one is summary slide. And then first notion was that after second dose vaccination, tetramer frequency gradually decreased up to 6 months follow-up period. But in the after third dose, it is stabilized. So it means that somehow there's some beneficial effect. And then to know the first dose effect, we also recruited for the first dose cohort, another cohort. So we couldn't obtain the longitudinal sample from this cohort because all of them, they got the Omicron infection after third dose vaccination. So this one is the age-matched different cohort, and they got this one is first dose, but this cohort is a very valuable cohort because they didn't get the Omicron infection before first dose vaccination. And then we followed this 6-month follow-up period. And then, again, we found that after first dose vaccination, the CD8 T cell response has been stabilized. And then, what happened if we directly compare this response? So we measured very simply interferon gamma ICS by stimulating them with a single epitope peptide. And then we found that, this one is the multiple frequency, pentamer frequency, sorry. So this one is the pentamer frequency. The second dose, third dose, and fourth dose, this was 6-month sample. So really, after third dose and fourth dose, significantly, there's increasing the frequency of the multiple positive cell. It means that it seems that repeated mRNA vaccination has some beneficial effect. And then not only this, the multiple frequency, also we did is the ICS, interferon gamma or TNF-alpha. And then also in this ICS, in functional study, after third dose and fourth dose, gradual increase of this, the interferon gamma secreting cell frequency, also in TNF-alpha. So far, in my data, it seems that also the polyfunctionality also increased. So far I showed that it seems that repeated mRNA vaccination, third dose and fourth dose, nicely boost CD8 T cell immunity in terms of the T cells frequency, also their function. Then, what happened in their differentiation, the status? After third dose and fourth dose, effect on memory T cell, after each dose gradually decreased, and then was stem cell-like memory cell. So after second dose, it increased, but not so much increased after fourth dose. And central memory cell, not so. But we focus on this one, the TEMNA cell, CD45RA TEMNA cell. It has been regarded as terminally differentiated CD8 T cell. And then after third dose, gradual increase. Even after fourth dose, more increase. So it seems that the terminally differentiated effector cell are increased after each vaccination. So this one is the comparison between 6-month sample, second, third, fourth dose. Really, especially this TEMNA cell increased after repeated vaccination. But stem cell-like memory T cell gradual decrease. But this was not so enough because the TEMNA cell, it is this one, CCR7 negative and CD45RA positive cell. So we know that naive cell, after activation, they become central memory and effector memory. So this one is main effector cell. But then if this effector cell more repeatedly the TCR stimulated, then they become terminally differentiated TEMNA cell. The traditional immunology, human immunology, we know that. But recently, it was known that the TEMNA cell is not just the kind of cell. There's some heterogeneity. The TEMNA cell includes not only terminally differentiated T cell, but also any other cell. So especially during the COVID-19 pandemic, it has been reported. So to know the heterogeneity in the TEMNA cell population, we performed the single-cell RNA analysis by using the SARS-CoV-2 multimer. By using this third dose and fourth dose, the recipient, we obtained the 6-month sample, and we stained them with the multimer, with DNA barcoded multimer, and then we performed this single-cell RNA analysis. And then, as you can see here, there are several clusters. For example, C4 is the central memory. So this one is also ADT, it means DNA barcoded CD markers included. So it's actually the CCR7 and CD45RA and CD127 is not mRNA level, real CD marker protein level. And C4 is the central memory, C3 effector memory. And we found that the cluster 0, 1, and 2, 3 clusters looks like the TEMNA population, CD45RA positive and CCR7 negative. But we found that their heterogeneity. It means that these two, the cluster 0 and cluster 1 and 2. And we found that most striking difference was CD127. It means that CD127 positive TEMNA and CD127 negative TEMNA there. And how different between those two populations. So this one is the two different distinct populations, and this one is the differentiation score. As you can see here, so this color, 127 positive one, the less differentiated cell, more precursor cell type. But CD127 negative one is a highly differentiated cell type. So we think that this one is more differentiated, terminally differentiated cell type. And also this one is the stem-like feature we compared. CD127 positive cell, they have high stem cell-like memory feature. But CD127 negative cell, they show the bottom level, the lowest level of this stem cell-like feature. In this way, by performing this single-cell RNA analysis, we found that in TEMNA cell population, there are two populations, CD127 positive and negative. And 127 positive is not terminally differentiated one. This one is more stem-like T cell. And CD127 negative TEMNA cell are real terminally differentiated one. Then, so we need to divide TEMNA cell into CD127 positive and negative. And as you can see here, especially this the terminally differentiated one, after second dose, third dose, and fourth dose, gradually it increased, the terminally differentiated one. And then, so somehow in human immunology, terminally differentiated cell type are overlapping with the senescent cell type. CD57 is well-known marker for the replicative senescent T cell marker. What's the replicative senescence? It means that CD57 positive cell has short telomere. So we confirm in our hands, really, CD57 positive cell has a short telomere, and beta-gal activity is well-known marker for the aged cell, senescent cell. Also, CD57 positive cell show the aged phenotype. And then, what happened? So really, in the CD127 negative TEMNA cell population, all of them, they are, not all, but almost all of them are CD57 positive cell. So if we gate CD57 positive, 127 negative TEMNA, really senescent, terminally differentiated memory T cell, after second, third, and fourth dose, really dramatic increase. And then if we directly compare the 6-month sample, then the significant increase after third dose and fourth dose. Then what's the functional implication of this terminal differentiation? At the beginning of this second part, I talked to you that in tetramer frequency, the pentamer frequency, and also interferon gamma ICS, it looks like that there are boosting effect. But then, at the same time, simultaneously, in the not only boosting effect, but also terminal differentiation, senescence, T cell senescence after second, third, fourth boosting vaccination. What's the outcome, functional outcome? So to know that, we examine the proliferation, the potential of this memory T cell. After second and third and fourth dose vaccination, we obtained this PBMC, and again, we stimulate them in the single epitope with this matched epitope with this pentamer, and then we perform the CTV analysis, CFSC analysis. But just at a glance, it looks like that. Oh, all of three memory population, it shows nice the proliferation, 90%, 98%, 95%, 98%. So very robust proliferation. So nothing to lose after first vaccination. So this one is summary data with the CTV low cell, immune proliferating cell. Really, nearly 90% of cells are proliferating. So really nice, the data. So we focus that this one is so much robust. So it is not so fair just comparing this CTV low. So in that way, so my graduate school student, he developed the idea. So we need to more meticulously, we need to examine this proliferation potential. So he developed two ideas. The first one is this one. Actually, in this case, the representative FACS plot, we measured this pentamer frequency ex vivo sample. And then 7 days, the peptide stimulation with low dose IL-2, there's some in vitro expansion, ex vivo expansion. Then he calculated how much fold expansion, the before and after 1 week, the expansion protocol. And then we found that roughly after second dose, 6 months memory cell, they shows roughly 20-fold change increase. After third dose, in average, 15-fold increase. After fourth dose, around 5 or 6-fold increase. So it looks like that still there's some huge increase, huge proliferation. Actually, their proliferation capacity is gradually decreased after third and fourth vaccination. And this one is more meticulous way of this proliferation, their capacity. Again, we look back again, this the CTV analysis. As you can see here, all of these three populations show nice proliferation. As you can see here, after second dose, this the main body, they underwent like 7 times the proliferation. But after fourth dose, main body, not at the left hand. Actually, the main body is around here, 4 times or 5 times proliferation. So in this way, we calculated mitotic index after first proliferation, second proliferation that way. So this the mitotic index. We calculated per each precursor cell, how many times for 1 week they expanded, they proliferated. So this one is mitotic index. Again, we found that second dose, the mitotic index was like 20, around 12, 15, and after fourth dose, around less than 10. In that way, it seems that with just the rough analysis, evaluation, nothing to lose after this multiple vaccination. But in this way, very detailed analysis, it showed that after first vaccination, there are more senescence and more terminal differentiation of the CD8 T cell. And then now I'm showing that this mitotic index very nicely correlate with this the proliferation, the fold expansion, the capacity. And then really this the decreased proliferation potential, it is connected to the terminal differentiation. To answer this question, we just analyze this one. From the whole donor, the 6-month sample, so this one is % of each donor, senescent cell and senescent terminally differentiated, the TEMNA cell, and their proliferation capacity. So nicely they show the inverse correlation. So it means that really after multiple vaccination, so tetramer frequency increase and then their interferon gamma ICS frequency increase, but they differentiate into more terminal differentiation, more senescent T cell, and then their proliferation capacity potential gradually decrease. So we don't know yet. So this is the end of my data. And yeah, right, this one is the end of my data. But we don't know yet. This one is due to mRNA vaccine or due to short interval, 6 months. So recently, Paul Kleinerman and then the Oxford group, they published in Science Immunology, in the short interval really enhance due to actually the more short duration of the vaccine-induced T cell. So I think that by increasing the interval of the vaccination, maybe we can reduce the terminal differentiation effect. But we need more experiment to do that. So this work was done in my lab by Dr. Mingyong Jung and Seungmin Jung. Yeah, thank you for your attention.
Very nice data. I have a curiosity that is coming from patients that now are doing therapeutic vaccination, where really the frequency of vaccination is every 2 weeks. What do you think? I mean, okay, we know that these are probably T cells that we want to expand for therapeutic vaccination that are different than the classical memory.
Okay, yeah.
But what do you think? I mean, also that it doesn't make a lot of sense.
So to answer to your question, so let me a little twist your question. So after presenting this talk, some people ask me about that. So what do you think? Is it better to get booster vaccine or not for this vaccine? But my answer is not just a single way. So now, currently, there's some outbreak. Then it's much better to get back to boosting vaccination because boosting induces immediate effector function, but not long-term durability. So in the situation of outbreak, we don't care about the long-term later, just immediate effector function. But there's no outbreak for the long-term maintenance. I will recommend do not boost vaccination. But in the therapeutic vaccination, usually we are thinking about immediate effector function, not the long-term, like 2 years later, no one cares about that. Yeah, so for the nice effector function, short duration is okay, I think so, yeah.
I'm here. Yeah, great talk. So quick question, but it could be a very complex answer. So can you comment on MFI, the Pentamers, in the different stage of the CD8 T cells?
Oh, actually, we didn't look at the MFI.
Oh, really? Okay.
Yeah.
Okay, thank you.
But yeah.